Hello, my name is Mariana Westergren. I'm from the Slovenian Forestry Institute, and I will tell you a little bit about the genetic structure and genetic diversity of Fraxinus angustifolia, so the narrow-leaved ash in the Mura-Drava-Danube Biosphere Reserve. The research was done in the framework of the Refocus project. We sampled the narrow-leaved ash trees from all 5 countries of the Mura-Drava-Danube Biosphere Reserve, from Austria, Slovenia, Croatia, Hungary, and Serbia. We sampled 30 adult trees and 30 saplings. These saplings were about 1 to 2 meters tall, and they have already gone through the initial steps of natural selection. Now in Slovenia, we sampled a little bit more trees because we already knew that the narrow-leaved ash hybridizes extensively with the common ash in this area. Also in Austria, the narrow-leaved ash doesn't appear in the Mura-Drava-Danube Biosphere Reserve, so another population was sampled still in the Danube region, but further to the northeast of the country. In addition, we sampled 20 common ash trees, and we sampled them in an area where no narrow-leaved ash grows. Just like common ash, the narrow-leaved ash is also highly susceptible to the ash dieback, a disease caused by a fungus. Now, one of the aims of this study was also to see whether we could find any correlations between the places in the genome of the narrow-leaved ash and the resistance to the disease. Therefore, we gave each ash tree a health score, 1 for a completely healthy tree and 6 for a dead tree. Now, on average, the trees were healthiest in Serbia, and they were sickest in Hungary and Slovenia. And this is probably because the disease came to Serbia later than, for example, to Slovenia, Austria, Hungary, and Croatia. After the leaves were sampled in the field, they were shipped to the DNA laboratory at the Slovenian Forestry Institute, where we extracted the DNA. We then sent the DNA to an outside provider, which carried out a spit analysis. The probes were selected based on the information of DNA regions which showed to be correlated with the resistance to the ash dieback in the common ash, and some of the probes were synthesized randomly. Now, after obtaining the results back, we filtered them, and then we did the analysis with the admixture and the Plink softwares. Narrow-leaved ash hybridizes with the common ash where they grow in vicinity, and we knew that this is the case for the Slovenian population. So the first thing we did is we checked for hybrids. The blue colors on this picture show the genome of the common ash, and the orange lines belong to the genome of the narrow-leaved ash. And we saw that in Slovenia we actually have quite a lot of hybrids. These hybrids were not at all common in the other countries of the Mura-Drava-Danube Biosphere Reserve. Looking at the genetic structure of the 5 populations, we can see clearly again that the common ash can be clearly differentiated from the narrow-leaved ash, and that we have hybrids in Slovenia. The second thing we can see is that the Hungarian gene pool pops up. It is quite different to the gene pools of the other populations. Secondly, we can see that the Slovenian and Croatian populations and partially Austrian populations are quite similar if we take out the hybrid component. And then the Serbian population in the next step also differentiates from both Croatian and Austrian ones, as well as from the Slovenian one. This information on the genetic structure is important for the management, for the conservation, as well as for the sourcing of forest reproductive material. Looking at the genetic diversity, it was estimated in two ways, as expected heterozygosity on the left panel and as a percentage of polymorphic loci on the right panel. Now, in general, they are quite well correlated, but what we can see is that the seedling cohort in the Serbian population had quite lower genetic diversity in terms of expected heterozygosity. Now, there might be other reasons, but we suspect that the main reason is the mistake with sampling, which in this and only this cohort was not carried out randomly. But in general, the genetic diversity of all the populations is quite high. As said at the beginning of the presentation, one of the aims was also to see whether we could find the correlation between the genome and the resistance to the ash dieback. And looking to more than 100,000 places in the genome, we found one such place, one SNP that might be correlated with the resistance to the ash dieback. Now, the frequency of this SNP is depicted in the orange color per population and per cohort. Now what we can see is that the frequency of this SNP is higher in the sapling population, so in the regeneration, which gives us hope for the future. On the other hand, we can see that in Serbia, in the population with the best resistance or the lowest infection rate of the trees, actually the frequency of this potentially advantageous allele is the same between the adults and the regeneration. While in Slovenia, the picture is a little bit different. This is the only population where the frequency of this interesting allele is lower in the seedling population than in the adults. We don't know why. It doesn't give us much hope, but it might be connected with the fact that we have a lot of hybrids between the common ash and the narrow-leaved ash in the Slovenian population. This was all on the genetics of the narrow-leaved ash in the Mura-Drava-Danube Biosphere Reserve, and thank you for your attention.